welcome you all to this year's Nobel Lecture Series. We have three speakers today with us who have most kindly accepted our invitation to elaborate on the significance of the groundbreaking discoveries in physics Nobel Prize this year. Professor Prateek from Department of Physics IISc will talk about the and physical evidence for existence of the supermassive black hole at the center of our galaxy, which in the words of Professor Andrea Ghez herself is observing something that you can't see. Professor D.N. Rao from Department of Biochemistry IISc will walk us through the journey of the development of a novel, extremely sophisticated yet simple method of gene editing by CRISPR, establishing yet again that the collaboration of chemistry and biology is the recipe for wonders. Professor Uday Ranga from Molecular Biology and Genetics Unit, JNCSR, will talk about the discovery of hepatitis C virus, the vaccine, and how it will save millions and millions of lives. We welcome questions at the end of each talk. Other than here, questions can also be posted at the YouTube comment section if you're watching this live by live streaming. Our coordinator Mani will convey your questions to the speaker. So let's get started, and I request Kirtana to introduce our first speaker. Over to Kirtana.
Hello.
Over to Kirtana.
Hello. Yeah. So, Professor Prateek Sharma from the department.
Audience are physicists, so I hope everyone will have something to take away from this talk. So let me get started. So this year's Nobel Prize awarded to these three scientists. The first half being given to Penrose, and I quote, for the discovery that black hole formation is a robust prediction of the general theory of relativity, and the second to Penrose, and I quote, for the discovery that black hole from half was shared between these two scientists, Reinhard Genzel and Andrea Ghez, for the discovery of a supermassive compact object at the clarified theoretical foundations of formation of black holes, and these folks performed experiments to show that they exist in reality in our observed universe. So the story of black holes actually start at the beginning of the 20th century, when two big revolutions took place in physics. One is, of course, quantum mechanics, and the other is the nature of space-time itself. And it's sort of, we all are aware, much more aware of quantum mechanics because of the numerous applications it has had in physics, in chemistry, and through chemistry part of the breakthrough. And this is the title of his 1905, one of the miracle papers of that year, which is in English translates to on the electrodynamics of moving bodies. It has nothing to do with space-time in the title, but the seed of relativity is in electromagnetism. This is what this paper is all about. So here I quote from Hermann Minkowski, the views of space and time which I wish to lay before you have sprung up from the soil of experimental physics. So these are experimental results. The nature of space-time is an experimental result, and therein lies their strength. They are radical. Henceforth, space by itself and time by itself are doomed to fade away into mere shadows, and only a kind of union of the two will preserve an independent reality. So basically what he's suggesting is that somehow the space and time get mixed up, okay? And this is the essence of the theory of relativity. Okay, so this was around 1905. Now, this was, so Einstein was thinking about how to incorporate gravity in the same sort of formalism. And in 1915, after a decade of a lot of hard work, he was able to come up with a formalism in which
लोग हैं
uh,
फर्स्ट दे देता है, साइन करवा लेता है, नंबर भी ले लेता है और चीजें नहीं बोला है
audience,
कि
please put yourself on mute if you can. Thank you. So basically, the upshot of Einstein's general relativity is that gravity is not really a force. It is the curving of space-time in the presence of mass and energy. So these effects are so important, but the question is, why are these not known? And the answer is, these are only perceptible when objects approach close to the speed of light, and when the gravity is very strong, in the sense of the escape velocity approaching the speed of light. So black holes is an outcome of gravitation, theory of gravity, and we can even think about it even in terms of simple high school physics. Now, simple question, right? So we are all familiar with this concept of escape velocity, that if you throw a ball up, if you throw the ball up with small speed, it comes back to you again after a short time. It's longer and longer to come back to you after reaching a maximum height. But if you throw a ball faster than the so-called escape velocity, escape speed, then this ball never comes back to you. It sort of escapes the gravitational influence. So people, thinkers like John Mitchell and Laplace, already in 18th century, were sort of contemplate on questions like this. Now, what if the escape speed of an object exceeds the speed of light? Right? If you have such an object for which the escape speed from its surface exceeds the speed of light, what this would mean that even light cannot escape such a strong gravity. Okay, or it will appear dark because not even light can come out of this object. Hence, this object is sort of called a black hole. So, already people much before relativity and modern concepts of gravity about these ideas. Okay, and so this is the expression for escape velocity: half mv square equals gm by r. Right. So, yes.
So there is some problem in the screen sharing. We can't see your screen and
I see. Okay, good. Thanks for.
I think somebody has taken control. Please do not take control. I would request Professor Pratik to share his screen again.
I'll do that. So,
yeah.
Has it been long since I was not able?
It has been just a minute, sir.
minutes.
We were just trying to figure it out.
Okay.
Yeah.
I will actually start from this escape speed again. Okay, no problem.
Please do not take control. Thank you.
Yeah. Please do not give control. Velocity. This is just square root of 2 gm by r, right? We have all learned this in our middle school and high school, right? So we can write. So if we say that the escape velocity reaches the speed of light, we can get a radius, right? We can write r equals 2 gm by c squared, and this is a radius of an object if the mass m is compressed to a size smaller than radius r, then even light cannot escape this object, right? So this object is so compact that outside world cannot see it. So this is like a simple idea behind a black hole, right? And if we plug in the numbers for these constants, we get that this comes out to be 3 kilometers for the mass of our sun, right? This radius is called the Schwarzschild radius, and this is 3 kilometers for the sun. So in other words, the sun has a radius of more than a million kilometers, more than it's almost 10 to the 11 centimeters. So if we were to compress it to 3 kilometers, then light, then the escape velocity would exceed the speed of light. So 3 kilometers is like the size of IISc campus. So all of our sun is compressed to the size of our IISc campus. So you can imagine the kind of densities that we are talking about. Okay, so if you have a black hole which is, so if you have an object which is more compact than this, then even light cannot escape it, and this appears dark. Okay, and this is a black hole. This is the classical or pre-general relativity notion of a black hole. Now, what about black holes in general relativity? So as I mentioned in the first slide, GR or general relativity is the correct theory of gravity, and Einstein sort of came up with it in 1915. So he derived uh, the equation for how uh, how gravity is produced because of mass in the universe, distribution of mass. He gave those equations. These were quite complicated tensor equations, and he did not believe that these equations can be solved that easily, which does not collapse to an infinite density. This is the question that was answered by Roger Penrose, and for that he was given this year's Nobel Prize. So in a paper in 1965 and subsequent papers and earlier papers, he introduced geometric methods, right? Geometric methods, for example, the notion of a trapped surface, which shows that the collapse to a singularity, collapse of a gravitating object, of a massive enough gravitating object to a single point of infinite density is generic. It's not a consequence of assumed symmetry. Okay, so this is the Nobel Prize winning work that was published in 1965. And in fact, this work makes use of the work of Amal Kumar Roy Chaudhary from Kolkata, who actually introduced something called Roy Chaudhary equations, which give the evolution of nearby trajectories in general relativity. So he used these concepts and polished them and applied them to prove that these singularities, these black holes, are generic features. So so on the left, I show something, a cartoon to show this development of a singularity because of a collapsing star. So y-axis, you can imagine that the time runs vertically, and the x-axis horizontally you have space. So you imagine at a given time there is a star, right? So there's a star which has some spatial extent, and it sort of runs out of its nuclear fuel, and it has a mass which exceeds this limit beyond which collapse is inevitable. So what will happen is all this star will compress in size with time, because the pressure forces cannot hold it against gravity, and it sort of collapses. And what I am showing here in this red, and this sort of goes till here, is something called an event horizon, right? An event horizon is a point from within which even light cannot escape. Okay, so the star is much bigger earlier on, like our sun, right? Light can escape very easily from the sun, but with the collapse, it eventually crosses the size limit, which is smaller than this Schwarzschild radius, corresponding to the surface, corresponding to the escape velocity exceeding the speed of light. Okay, so once that happens, once this matter is compressed to smaller than the Schwarzschild radius, nothing comes out of it, and moreover, no forces can prevent its collapse to a singularity, to an infinite density. Okay, and here, so this this orangish pinkish color thing shows the collapse of matter. These cones, these double cones, show the trajectory of light. So if you are outside this red surface, light can travel outside, right? Light can go out and travel to an observer outside. But as soon as this surface which emits this light is inside this event horizon, all this light is directed towards this time axis. Nothing can come out to an observer outside. So this is the development of this event horizon, and this is the simplest trapped surface. So here, right, this white circle here shows a trapped surface. So this matter, which was sort of which was not as compact earlier, sort of becomes more and more compact, and at some point it is confined by this trapped surface, out of which nothing can escape, not even light can escape, and within which the formation of singularities or black holes is inevitable. So this is shown in a very, very simple setup, spherically symmetric setup. Penrose's contribution was to generalize this idea to general collapse with angular momentum, with asymmetry. So that's why this was a big achievement, because what it says is that formation of black holes and singularities are generic. They do not depend on this assumption of excessive symmetry. Now, what about actual realization of black holes in astrophysics? So here is actually a schematic of what happens to stars when they run out of their nuclear fuels. So what happens is that the fate of stars depend on their initial mass. So if the star's mass is less than 8 solar masses, so all stars will swell up at some point, but eventually such a star will leave behind a very compact object called a white dwarf, right? And this white dwarf has a mass less than 1.4 solar masses, which is known as the famous Chandrasekhar limit. These stars are supported by something called the degeneracy pressure. So this is pressure due to electrons which have been packed close together, right? We know that electrons are fermions and they don't like to be very close next to each other. So this act of gravity which tries to push it together results in a pressure which can handle only compact objects which are less than 1.4 solar masses. If the star's mass is from 8 to 20 solar masses, you form something called neutron star, which is also degeneracy pressure supported, but in addition also has nuclear forces providing the pressure against gravitational collapse. Now, if you have a very massive star greater than 20 solar masses, then you have a remnant which is greater than 3 solar masses, and it will collapse to a black hole. Okay, so this is what the fate of stars is. So our own sun will, after 5 billion years or so, form a white dwarf. Okay, so in astrophysics, in reality, we have black holes of two kinds: stellar mass black holes, which have mass going from 5 to 100. These are ballpark numbers, to a few hundred solar masses, and these are detected in X-rays, and more recently in gravitational waves. I'm sure a lot of you have heard about gravitational waves emitted by observed by LIGO over the last several years actually. Now, these are stellar mass black holes formed from the death of stars, but there are other black holes as well in astrophysics which are called supermassive black holes. These black holes are present at the centers of galaxies, and their masses range from 1 million to 1 billion solar masses, right? So these are two images of such black holes. This is something called a quasar. So this is an optical image taken by Hubble Space Telescope, and it is showing a very, very bright point source. This point source is so bright that it outshines the whole galaxy that it sits in, right? When we normally look at a galaxy like this one, you see a lot of stars, diffuse light due to a lot of stars. But in this case, this point-like object is so bright that it outshines everything else. You cannot see the other stars in this galaxy, just like you cannot see other stars in daytime because of the glare of the sun. So, these objects are so powerful and so compact in space, and they show so much variability in time that people think that these are powered by very massive black holes, right? Million to a billion solar mass black hole. So this is an optical image. Image on the left shows a radio image which shows these magenta bubbles and these jets. And in the background you have an optical image. So you see these things, these plus signs, crosses, these are stars of our own galaxy. Just ignore these. But you see this galaxy, sort of a smudgy collection of stars. So this is also a result of a supermassive black hole powering these radio emitting jets of plasma. So these were observed starting in 1950s, and these bubbles, these radio bubbles, occupy such a huge volume. Like this is the size of the galaxy at the center in comparison. So the extent of these bubbles is more than 10 times the size of this galaxy. So these are some of the evidences for the existence of very massive black holes at the centers of galaxy. And now almost everyone agrees that there is a million to a billion solar mass black hole at the center of almost all galaxies. And these fireworks in radio and in optical are powered by gravitational energy because of this strong gravity of black holes. And this energy is dissipated in the surrounding matter and produces this light. So the second part of this Nobel Prize was given for unambiguous detection of black holes. So the question is, how can we see black holes as not even light comes out of the event horizon, right? And the answer for this question is that we don't see the light directly from the black hole, but by observing fast moving objects around it. For example, fast moving gas and stars around the black hole. So here are two examples. So this is the Nobel Prize winning work showing orbits of stars around a dark object somewhere here, which is the location of a very massive 4 million solar mass black hole in the center of our Milky Way galaxy. These orbits are traced over a period of more than 10 years, 1995 to 2008, and you can actually see these orbits, which are almost like Kepler's elliptical orbits projected on the sky. So this is the Nobel Prize winning work. I'll talk about this more. Some of you may have also heard about this image of a black hole that was recently announced like a year or a year and a half ago by this Event Horizon Telescope. This is a radio image of a nearby supermassive black hole in a massive galaxy M87. And what it shows is an image obtained by radio telescopes spread throughout the Earth, right? So that you can combine signals from all those and reconstruct how this radio source look like. So you can actually see like a ring feature and darkness at the center. So this is a characteristic signature of a black hole for which the matter around it is emitting, but the black hole itself does not emit any light. So I'll not talk about this. So this is the image of the center of our Milky Way galaxy in optical. So this is the galactic center, and you see it's very, very dusty and opaque, right? If you look at this part of the sky, you cannot see because there's a lot of dust on the way, right? Whereas if you look at this part of the sky, it's not as dusty. So because of the dust, it's very difficult to see in the center of our Milky Way to see whether there is a black hole there. So this Nobel Prize winning work was actually about imaging the center of our Milky Way galaxy to see the signatures of a supermassive black hole. So this work cannot be done in optical light or UV ultraviolet. We have to go to infrared, because in infrared, the dust emission is not that severe and you can see through this material in infrared. So let me play this movie. This movie is quite nice. It shows like zooming in on the galactic center. You go across sort of these various structures that appear and because you're sort of starting at 10 kpc and zooming on to a size of almost 0.01 parsec. So parsec is the length that's used in astrophysics. 1 parsec is like 1/3 of a light year. So the point is, we are talking about uh, 0.01 to 10 to the 4. So 6 orders of magnitude in scale here, right? We are talking about a scale of 6 orders of magnitude that we have to zoom in to see what's happening in the center of our, let me pause this movie. Okay, so now what we have done is, actually, let me play this again. So we are actually just zooming in. So these are the constellations of Scorpius and Sagittarius, and the galactic center lies in the Sagittarius constellation. That's why it's called Sagittarius A star. And you just see stars. There is nothing special at the galactic center at these wavelengths. It could have been here, or anywhere else. But we know that there is something special here from radio telescopes. There is a very, very bright compact object at the center of our galaxy. So this is not a radio image. This is a composite image in optical and infrared, where the galactic center is not at all any special. Okay, now this, right, so let me pause it here. Now, these are actually the highest resolution images obtained in the galactic center. And these white things are the location of infrared emitting stars in the galactic center. And what you should notice is that these stars will move around as I play this movie. So, yes, so you can see this motion of these stars. So this is actually a real observation taken towards the galactic center from the Max Planck MPE group, and you see this motion of stars around a dark object which we cannot even see in this. So you see this object, try to follow this object, and you will see, right? So it moves very, very fast, and you can actually resolve the motion of this object. Now, this actually is an animation. This is not a, oops, let me go back. That was an animation, not a real, this is not a real astronomical image. Whereas these white blobs were, these were real images of images taken in infrared in the galactic center. Now this is an animation which shows the trajectories of these stars around an invisible object here, which is very, very massive. We know that this is so this is the black hole around which these point particles, these tracer stars go around, and these trajectories are all, occupy all the angles and orientations in space. Unlike the solar system planets, they are all in a plane. These stars are actually 10 solar mass type stars, very massive stars, and they just sort of trace out this trajectory. So they are able to trace out the space-time or the gravity around this black hole. So this is actually a result of more quantitative result of one of the orbits of those stars called S2 star. This is the orbit traced out for this S2 star over a period of about 20 years. You see, this is 1992, and you just sort of map it out on the sky. And in 2013 you come back here, so you complete a full orbit in about 17 years, and this is more than that. So you actually see like a projected orbit, a projected Kepler's orbit on the sky around an invisible object. This is Sagittarius A star. Its location is marked by the radio source that we already know. So you see, this is a projected Keplerian ellipse on the sky, and this massive 4 million solar mass black hole is sitting at one of the one of the foci of this orbit. Right? This is very, very simple, very, very simple mechanics. This is orbit of a test particle around a massive object. So this is the orbit. This is RA and DEC. These are basically two angles in the sky, and it sort of traces out an ellipse, a projected ellipse. This is the line of sight velocity measured from the Earth, basically the Doppler velocity measured as a function of time. So when this star S2 is going away from us, for example here, when this star is going away from us, you see velocity which is red shifted, and when they start approaching us, you see velocity which is blue shifted. So you see this particular star reaching velocity as high as 4,000 kilometer per second, or at least thousands of kilometers per second. You never see such fast moving objects or stars in astronomy. So these fast moving object means that these are going around in a very, very strong gravitational field. And you can combine this motion in the sky and this Doppler velocity to obtain the mass of this object and the distance of this object. And the mass comes out to be around 4 million solar masses, and the distance is 8 kiloparsecs, which is the distance to the center of our galaxy. Now, what this shows is the enclosed mass. So mass within a given radius as a function of radius from the center of our Milky Way galaxy, right? So the farther you are from the center, the lower resolution you need. So these points were available like several decades ago. But to get to, so this point corresponds to this S2 star, which is very, very close to the supermassive black hole. So the fact that the S2's orbit is perfectly described by a Keplerian orbit means that there is no deviation from a point mass within that radius, right? So there is a very, very compact, massive star at the center. So if you obtain a limit on the density of this region, density of stars in this, density of matter in that region, it comes out to be 10 to the 17 solar masses per parsec cube. In comparison, the density of stars in sun's neighborhood is 1 solar mass per parsec cube. This is 17 orders of magnitude higher density of stars in this of mass in this region. And this dense matter cannot be anything but a black hole, right? If you had, for example, a star cluster, it cannot be so dense. It would sort of dissipate on a very short time if it was so dense. So in comparison, these are star clusters, and their density is like a few million solar mass per parsec cube. So this work was obtained from the motion of stars, and strongest constraint come from this S2 star, which goes as close as 100 astronomical units. 1 astronomical unit is the distance of the sun, distance between the sun and the earth. And this is still far away from the Schwarzschild radius of this black hole, about 2000 Schwarzschild radii. Now, this work had to overcome some technical problems. So what I'm showing here is like image of the surface of the moon. And what you see here is the jittering of the image, right? And this jittering of the image is because of atmospheric turbulence, right? This is another example. This is a point source which gets spread out because of the turbulent atmosphere. And in order to image this orbit of the black hole, of the stars around black holes, we need to overcome these effects, okay? Because we cannot get very high precision orbits if we cannot overcome this atmospheric jitter. We cannot achieve diffraction limited imaging of 0.01 arc seconds unless this is mitigated. And this is partly the reason that telescopes are situated at an altitude, because if you go above the atmosphere, the turbulence is to some degree mitigated. So this is a technical challenge that has to be overcome, that had to be overcome in order for these people to detect the motion of stars around supermassive black holes. So here is a demonstration. This is the image around the region of Sagittarius A star, the galactic center black hole, without correcting for this atmospheric turbulence. You really cannot, resolve any of these stars. They are very fuzzy, right? Now, if you use something called speckle imaging, which is like very, very quickly taking photos of that region, so that the atmospheric turbulence doesn't have time to distort this image, and combining them later, you can obtain such a quality image. So this is much better than this. Of course, you can see these stars, and you can hope to sort of map out some of the orbits of these stars. So this is speckle imaging, but even better than that is something called adaptive optics. These are images obtained from a variety of adaptive optics, and you can actually see the stars very well. And because of adaptive optics and speckle imaging, people were able to obtain these nice looking elliptical orbits. So the idea of adaptive optics is illustrated here in this cartoon. So you have some incoming wavefront of light, and the atmosphere distorts it. Okay, there is a wavefront sensor which sees how this atmospheric motion has distorted the wavefront, and it sort of moves the mirrors of the telescopes a little bit by some sort of piezo sensors, and it compensates for this distortion produced by the atmosphere, and it produces a nice looking wavefront again, and you can actually obtain a diffraction limited imaging, and because of this you can actually monitor these stellar orbits that I was showing. So this was the technical thing that had to be overcome.
Sir,
Right.
I'm sorry to interrupt. We are running a little short of time.
Sure. So I'm actually at my last slide.
Yeah,
So,
okay. Thank you.
So this is my conclusion slide. So to conclude, black holes are a robust prediction of general relativity, this theory of gravity. So this was so to mathematically demonstrate this and to develop the techniques which showed that singularities of black holes are generic predictions of GR. Roger Penrose was given half of the prize, and by monitoring these orbits of stars around this invisible object, Genzel and Ghez were awarded the second half of the prize. These,
Ask comments.
So the existence of black holes is now verified by independent observational probes. This is a work of 30 years. This observational work, took a long time, and there's a lot of technology evolved. For example, this adaptive optics to correct for atmospheric turbulence, and the future is very bright for detecting and characterizing black holes. You must have heard of Event Horizon Telescope and other big telescopes coming up. And black hole mergers, of course, this is the other Nobel Prize winning work on black holes from 2 years ago, I think, which showed that black holes are real and they have very important role in astrophysics. So with that, I thank you. Thank you.
Thank you for that engaging talk, sir. And the floor is now open for questions. I see that Mr. Rajarshi Bhattacharya has their hand up.
Yes. So a very nice talk, sir. And I'm a chemistry student, so I don't know much about black holes and physics. But I have this one question that if even light cannot escape a black hole, I mean, what happens to the information that gets inside a black hole? I mean, information cannot be lost. Say, for example, you put in any electromagnetic wave, and it might get scrambled, but the information has to stay in some form. So what happens to that?
Right, that's a very good question, and that's actually an open question which people are debating about. And I think there was also a very famous bet on this between Stephen Hawking and I think Kip Thorne, or one of these people, one of these relativists. So for practical purposes, for practical purposes, yes, information, the entropy has to always increase and all that, but this this work was really was not really answering these aspects of black holes, but more the observational and the general relativistic aspect. So there is also a quantum aspect of black holes, and there is something called Hawking radiation and concepts like this. So what you're asking is much more related to that. and I'm afraid I'm not an expert on that.
And
So,
sir, another question is that probably in 1939 itself, when Einstein was about to prove his theory of relativity during an solar eclipse, so then he used uh, the concept of gravitational lensing to uh, to 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 determine the position of the stars during the eclipse. So,
right.
if that is the case, then why weren't black holes detected much earlier? I mean, if the concept of gravitational lensing had been there for so long,
Okay, let
then
me answer
why?
this question. I can answer very well. So, gravitational lensing, even Earth would lens objects. Gravitational lensing does not require strong gravity. Lensing is extreme when the gravity is strong. So you don't require a black hole to lens background objects. Even weak gravity can sort of bend the lights to some degree.
Yes, I mean, isn't the extent of lensing be measured quantitatively?
Right. So there are like black hole lensing measurements too. That's correct. Yes. But they are extreme, they are not like the lensing of background stars observed during the solar eclipse of 1919, led by Arthur Eddington. It's not
Yes.
that. So that's a different thing. That's very small lensing, by 1 arc second or something like that. Very tiny. And that does not require a black hole. Even weak gravity is enough to do that bending of light.
Okay. Okay. Thank you, sir.
Mhm.
Okay, since we're running a little short of time, we'll quickly take two more questions. Nagraja sir?
This is a fascinating talk, Pratik. Uh, I have been always fascinated. I wish I am not a biologist. I should have studied a little bit of astrophysics. It's a real. Thank you very much. The the couple of quick questions. This white dwarfs and neutron stars, they cannot fuse and they can also cannot collapse into the black hole.
Right. So white dwarf,
Or neutron stars.
or
Neutron stars fuse.
yes, yeah, yeah, yeah.
And that can lead to, could they also collapse into the, like any other star, they can also collapse?
Right. So white dwarfs basically are equilibrium structures like stars, right? In a star, stars does not collapse because there is, the pressure is maintained by nuclear burning at the center. Even though there is energy lost from the surface, in the same way, white dwarf is an equilibrium object. It can stay very, very long. It is like it has an infinite lifetime. In the same way, a neutron star can stay very, very long because they are nicely supported against gravity by other forces. Whereas a black hole is an object, black hole forms when no other object can prevent this eventual dominance of gravity. So, black holes are objects where gravity is supreme. No other forces of nature can stop it.
Okay, but then even the black holes, the super black holes, then they can collide. So the possibility exists that two black holes in the center of two galaxies, in principle, when they are unregulated or whatever it is, they should be able to
Yes.
collide, and two galaxies will become one.
Yes, absolutely. You see you see mergers of galaxies very routinely. Mergers of black holes, supermassive black holes are more difficult to observe, but LIGO, you must have heard of this gravitational wave detection, this Nobel Prize to LIGO team on that. So that basically is looking at merger of these stellar mass black holes, not the supermassive ones from the mergers of galaxies, but there is something called LISA. This is a space interferometric experiment which will detect supermassive black hole mergers of the kind that you're talking about.
Okay. I think I will save for the sake of time. I think I need to talk to you something more about these things and also universe expansion some other time, maybe
Sure.
after. Thank you.
So just a follow-up question of Nagaraja sir. So after billions of years, Andromeda will collide with Milky Way, right?
Yes, I know that.
So
That was told, and it will
Yes.
not be. Yeah.
if that is the case, then would the two black holes just merge together, or will they be rotating each other? Because both are gravitational objects, and they both can just orbit each other.
Right. So you have a very good question. Now, it really depends. Mergers of galaxies are more common than mergers of black holes, right? Because the size of black holes are tiny compared to the size of galaxies, right? It's the cross section.
Yeah.
So yes. I mean, the people have estimated rates of black hole mergers, not necessarily the merger of our own Milky Way black hole and the Andromeda black hole. But the rate is reasonably high that people are trying to invest billions of dollars to put this interferometer in space to measure these things. Yes.
Thank you for the discussion. In the interest of time, we'll move on to the next speaker, and I request the others who have their hands up to please drop their questions in the chat box, and maybe the speaker could have a look at them there.
Sure.
Thank you.
I'll stop sharing then.
What should we? Yeah.
I request Sheetal to take over.
Yeah, I'll see. I don't see. Oh, it's there. Can you? Yeah, it's in the middle. He has to call now.
Hello. Am I audible?
Yes, yes,
Yeah.
go on.
Thanks, Kirtana. Now I'd like to welcome our next speaker, Professor Uday Kumar Ranga from JNCSR, who will be delivering the next talk for today's Nobel Lecture Series 2020 for the category of Physiology or Medicine, which was jointly awarded to three scientists, Harvey J. Alter, Michelle Houghton, and Charles M. Rice, for the discovery of a novel virus that is hepatitis C virus. They have made a decisive contribution to the fight against blood-borne hepatitis, a major global health problem that causes cirrhosis and liver cancer worldwide. Professor Uday Kumar Ranga obtained his PhD from School of Life Sciences, JNU, in 1990. His postdoctoral training involved working with HIV at the Center of Biologic Evaluation and Research, FDA US, from 1991 to 93, and at the University of Michigan from 1993 to 97. He established an HIV laboratory at JNCSR in 1997. His laboratory is one of the few in India that focuses on HIV pathogenesis, immune response, vaccine design, and technology development. Professor Ranga has been working with HIV for past 25 years, during which he also bagged two patents. He is a member of the Trainee Selection Panel, AIDS International Training and Research Program of Albert Einstein School of Medicine, New York. He is also a member of various other national and international societies such as American Society of Microbiologists, International AIDS Society, National Immunology Society, and Molecular Immunology Forum. With this, I'd like to welcome Professor Ranga. Sir, you can share your slides now.
Yes. Thank you and good afternoon to all. I'm just going to start my presentation first. All right, I started from the other end. Doesn't matter. Let me get my cursor. Oh, sorry. Wait. Full screen. One second. I'm getting my cursor. All right. Good afternoon again to all, and thank you for this opportunity. Can you all hear me clearly?
Yes, sir.
Yeah, great. And so this is our Nobel Prize for in Physiology of Medicine in 2020, given to three scientists: Harvey Alter, Michael Houghton, and Charles Rice. See, these three people made three important contributions at very different phases of this discovery. Alter for initial leads, and Houghton for molecular identification of the virus, and finally Charles Rice for establishing the causality. We are going to look at each of them in detail, but this is how I organize my talk into four parts. Introduction to the liver and hepatitis. I mean, it's all about liver and hepatitis infection of the liver, and a brief introduction to virus and HCV especially, and how HCV was discovered and why all this this celebration about this discovery, and finally where are we heading in future. Okay, introduction. Here is the liver. We all know. I don't want to take much time on this. It's a very vital organelle, and it controls more than 200 functions, including immune reactions. Does so many things. So I since it's general knowledge, I will skip this slide. And hepatitis is Greek word for inflammation of the liver. I mean, this doesn't have to be always by microbial organisms, as we see in the next slide, but these are certain clinical symptoms: appetite loss, vomiting, fatigue, pain, so on and so forth. And as I mentioned, it doesn't have to be always, infection that causes hepatitis. It could be also chemicals, radiation, and also autoimmune responses when they go haywire. But we are going to focus today mainly on this part of this viral hepatitis called the problem of the virus, jaundice caused by viral infections. So long time ago, people already knew, I mean, beginning of the 20th century, that this hepatitis infection can be because of two important reasons. One of them they call infections, epidemic hepatitis. The other is serum hepatitis. In other words, this is blood hepatitis. This is not blood hepatitis. So this is mainly transmitted through contaminated food and water. The infection is of very short duration. It doesn't cause serious illness, and usually it goes away very quickly, as opposed to serum hepatitis, which is in the blood. It is transmitted through contact with the blood and blood products, long incubation period, chronic illness, more importantly, liver failure many times. Okay, so these two are very different. And there are overall, we have very well characterized 5 viruses that cause this hepatitis virus due to viruses. There are additional viruses, but these are the 5 very well understood viruses. So there are 2 of them under this contaminated food viruses. One of them is called hepatitis A virus, short form HAV. And hepatitis E virus, we have ABCD. So AE caused this food contamination. The rest of the 3, hepatitis B virus, C virus, and D virus, they cause blood infection. So they belong to different families. HEV and HB belong to the same family, and except for HBV, that is a DNA virus, all other 4 are RNA viruses. Okay, so these are a few informations about regarding all these 5 viruses, but our focus today is going to be on this. But before we go there, we are going to look at a few more things. Okay, here due to chronic manifestation, serum hepatitis is more hazardous. So this is much more dangerous than this one. So we will see why that is. And this is a small list, a very brief list of so many Nobel Prizes given out for discovery of various infectious diseases, tuberculosis, malaria, so many. There are also viruses. The recent discoveries mainly in 2008 for human papilloma virus and also for human immunodeficiency HIV. And this year it is for HCV. So why there is so much importance attached to pathogenic organisms? So before I address that question, we'll briefly look at viruses and their classification. You all already must be knowing that we can classify the life into 3 primary groups according to phylogenetic, means genetic relationship. They are bacteria, archaea, and eukaryota. We all come, plants and us come under this, but viruses belong to a 4th group, right? So we are going to look at these viruses today. So what are viruses? The main important quality of viruses is they lack organelle structures. There is no ribosome, no nucleus, no nothing. Therefore, they cannot make their own vital proteins. That is the reason they need living hosts, and therefore they are obligate parasites. And the second important quality is either their information is either stored in DNA or RNA. Based on that, we can actually classify them into RNA and DNA viruses. They come in many shapes and morphology, sizes and all. We'll skip that. But one estimate tells us that at least 1.67 million viruses are yet to be discovered. Even if one of 1,000 is infectious, we are talking about something 10 to 10,000 pathogenic viruses that are going to emerge in the future. This is an estimate, and this estimate cannot be too far from reality. Okay, viruses are really small and they come in multiple shapes, including rabies virus like bullet. This is a bacterial virus. So here is the human cell, something like 100 microns, and here is the virus, some 100 to 1,000 times smaller. So they are very tiny. They are in the nanometer size. And classification, there are multiple ways you can classify viruses. One of these popular ways is based on host classification. Bacterial viruses, plant viruses, animal viruses are based on the nucleotide they contain. RNA viruses, DNA viruses, double stranded RNA, single stranded, all kind of classifications. But for pathogenesis, in the field of pathogenesis, this Baltimore classification is much more popular, which takes into account the primary genetic material that virus contains, single-stranded DNA or double-stranded DNA, double-stranded RNA, single-strand RNA, two different forms, positive orientation, negative orientation, and reverse transcript, reverse RNA converted to DNA viruses, and so on. There are 7 important groups identified based on this genetic material and replication intermediates. And this is a very useful classification for those people who study pathogenic properties of RNAs. And here is a basic fundamental structure of hepatitis C virus. It's a small, tiny virus, enveloped. It has, these receptors sticking on the surface. It is very small, nanometer size, 55 to 65. And it contains an RNA that is positive sense. What do you mean by positive sense? This RNA itself can directly code for proteins if it enters mammalian cell cytoplasm, and it belongs to Flaviviridae. And these are some few statistics. They look like numbers, but they are real grave figures. About 700 million cases globally per year. Total. And there are this many deaths every year. And hepatitis C, biggest problem with hepatitis C is liver failure. Okay, this is the life cycle. Like many viruses, it attaches to a receptor on the cell surface. There are many receptors identified. It enters through endocytosis into the cytoplasm. It never goes into the nucleus. It stays only in the cytoplasm, and it uncoats. Then a part of this RNA directly translated in the endoplasmic reticulum, and RNA is replicated. It has the machinery to copy RNA to make multiple copies, negative and again back to positive strand. And all of that again reassembles. They make into an exosome, and that is released. So the whole thing happens in a few hours, and that completes the life cycle. And HIV pathogenesis, it is mainly transmitted through injection, drug use, that is, people who use drugs, or through sexual contact, or through blood transfusion. So somehow this virus should get into your blood, or you should be contacted the liver, that how looks like very healthy in a normal human being. But once a person is infected, there could be multiple stages of pathogenesis, chronic hepatitis, cirrhosis, and liver already stopped functioning, and finally death. So that is what the virus does to the liver. And all the regions of the world are infected, contain HCV to some extent or the other. Some of them are heavily infected, some of them are lightly infected, but all of them are infected. Very few regions are actually free. Okay, now with this introduction, we are now getting into how HCV was discovered and why it is so important, why so much importance attached to this. From the scriptures, we understand that actually starting nearly even before, Christ, 400 BC, the first description of hepatitis is, we know from the statement of Hippocrates, where he stated the bile contained in the liver is full of phlegm and blood and erupts. After such an eruption, the patient soon raves, becomes angry, talks nonsense, and barks like a dog. This was one of the earliest recorded statements of a medical person of hepatitis. That is where the quest starts about hepatitis. And now this is the discovery of hepatitis A. I want to show you how A was discovered, how B was discovered before we reach C. Right here, the A virus is, as I said, is transmitted to contaminated food and water. So initially, scientists started looking for this hepatitis A virus in the blood, but actually this virus doesn't sit in the blood. So finally, later, scientists understood that the virus A is mostly in the feces. That is very hard to find this blood. So Feinstein in 1973 identified this viral particle in stool of one of the volunteers, and finally, they did a lot of work, and they even developed a vaccine for HIV. And very importantly, this vaccine protects up to 90%. That immune response established is very long lasting, up to decades, 25 years to 30 years. So it is one of the very protective vaccines that we have for hepatitis A. About 1970s that happened. And comes hepatitis B virus discovery, which won Nobel Prize in 1976. So here, this was work of Dr. Baruch Bloomberg. He identified a new lipoprotein in some people that are suffering from liver problems. And that lipoprotein is called Australian antigen because this antigen was first identified in an Australian Aborigine patient. And then they identified that this antigen can recognize antibodies in multiple patients who have this hepatitis. And one of the laboratory technicians was infected with this hepatitis virus. By the time they knew it was a virus. And Bloomberg suspected that this is probably because of this Australian antigen containing virus. And finally, they developed immunological tests for HBV screening. The important thing is there are blood banks, there is contaminated blood, so these tests are important. And he also developed a vaccine for HBV, and for these contributions, he won Nobel Prize in 1976. But even after the screening and eliminating some of these contaminated blood samples, still a large number of blood samples are still contaminated something that is still causing hepatitis. Now the problem here is if it is not hepatitis B virus, what is it? So this is where the story of HIV, HCV starts. So now we have two viruses very well understood. One is hepatitis A virus transmitted by contaminated food and water, and hepatitis B virus transmitted by blood and other body fluids. So people started using tests to screen blood samples that prevented only 25 to 50% of infections, but still there is contamination continuing, and questions arise to understand, is this a new virus or a different virus? This is where our real story begins. In 1970s, so Dr. Harvey Alter, he was working at National Institute of Health in US, and he demonstrated during his work that HBV screening prevents only up to 20% of cases. That means there are 80% of cases that are still carrying something that is not HBV, which he called non-HBV virus. And also these infections, clinical manifestations were different. So his main contribution comes from this. Here is his real contribution. What he did was he developed a primate model of infection, chimpanzees. So this virus or the contaminated blood samples can be injected into the chimpanzees, and those chimpanzees can develop the same morphology, pathogenesis in the hepatocytes, liver cells. And he did anonymous characterization of this infectious agent, and finally he was the person who identified that it's an enveloped virus of this size. So his contributions primarily concluded that at least there are two different viruses that are causing hepatitis. We already know one is HBV, that means there is one more, that is HCV, but this is the experiment what he did. We already know that the blood, if it is gone to other person, can contaminate and he can develop hepatitis. So what he did was, Alter did was, collect this serum from this person who is infected, inject into the chimpanzee and generate all this pathogenesis. Therefore, prove that there is a virus in this person which is not HBV, and it can recapitulate all the clinical symptoms and disease in the chimpanzee. That is his main contribution. That is where his work mainly stops. But the problem here was he could not identify what was that virus, though he identified the properties. Therefore, here comes our second hero, Michael Houghton. He's worked after 10 years in a biotechnology company called Chiron. And it was not him alone. There were three other collaborators. All of them actually together contributed. And these are all the people at CDC and Chiron. They contributed significantly to his work. And actually, he's Houghton at his when he was receiving this award, he said this: "It is a sweet to get them, that is the prize, but it is a better taste because they don't acknowledge the whole team directly. Without their input, I doubt I would have." Contribution actually, Kuo and Chu, they actually designed this experiment, what he used to identify this. We'll see how he did this. So this is his main contribution. What he did was, they initially started a classical screening assay and they screened tens of millions of bacterial colonies without any success. That is the time Kuo and Chu suggested a new immunoscreening approach. What is this immunoscreening approach? You, instead of looking for this virus, go to the clinical samples, plasma samples from the patients, infected patients, convert that RNA into cDNA and clone that into bacterial plasmids. So by doing this, you can grow these. If there is a virus there, if there is a viral RNA there, that will be cloned into these bacterial plasmids and I can amplify into millions and millions of copies. Here also other RNA, host RNAs will also get amplified, but I hope that one or two of them will be coming from the RNA. Now, this is a huge library, probably one or two viral plasmids being there. Now I screen all these colonies using antibodies coming from this patient who are infected with a non-B hepatitis virus. So I have millions of plates and millions of colonies here. Now I use these antibodies. Those antibodies detect only those clones which contain viral sequence there. That is what they did. So when they screened like this millions of bacterial colonies, finally they ended up with one important clone, what they called 511. It's a bacterial colony that contained 10,000 nucleotide containing viral sequence. Now they know that they have the virus on their hands. So this is the main contribution of Houghton's group. The work of Alter and Houghton, now these two contributions collectively establish a link between non-A, non-B hepatitis and HCV infection. But the problem here is still something is still missing. That is, they haven't established causality. That means they haven't yet shown that this is the virus and can infect this. That they haven't shown. That is where we get our third hero. Somebody switched on the mic. Please switch off. Okay, so before we go forward, this is what I said. They could not establish.
Hello? Sir, yeah. Hello. Professor Ranga, please unmute yourself.
Okay, can you hear me now?
Yes, sir.
Okay, let me back to my um, where is the view F5? Okay. Okay, this is where we stopped. Okay, Robert Koch, as you know, he is the person who gave us these guidelines. This is a must to identify a pathogenic organism. What he said was a microorganism must be identified in all individuals affected by a disease, but this should not be in the healthy individuals. Condition 1. Condition 2. The microorganism must be isolated from the diseased individual and grown in culture. Condition 2. Condition 3. When introduced, so I have the microorganisms in my hands, in my culture, they should be put back into the organism or an animal, and that should cause disease. Condition 4. from this infected animal, I should be able to isolate this organism, pathogenic organism, and that must be identical microorganism. So these four must be followed, and that was accomplished by our third scientist, Charles Rice, in 1980s and 90s. He was working at Washington University. So what he did was, he identified that many people tried this. They wanted to inject this viral RNA into the animals to recapitulate the disease. But Rice understood that the virus which is identified like this has some missing regulatory elements. So actually he first fixed that problem. He re-engineered the virus such a way that the missing things are added and then injected into the primate, into the chimpanzee. Now he could produce infection in these animals, and these animals develop clinical signs and hepatitis, and virus was found in the blood for several months. So his experiments finally concluded or provided enough evidence to show that yes, indeed, HCV is non-A, non-B hepatitis virus, which can produce all human infections. So this is the main contribution of all these three scientists, which won them the Nobel Prize. Now, what is the impact of this research? Because of the research, now today we have multiple tests available. Now we can screen all the blood samples and eliminate infected blood with both HBV and HCV. So blood is very safe today. And number two, we have very powerful medicines, especially for HBV and HCV. And these drugs can prevent up to 95% of infections or disease development in patients. But unfortunately, still we do not have a HIV vaccine. Therefore, the story is not yet complete. Now, this is the problem here. So we have HIV infection causing 114 million cases per year, or HBV causing several million infections, including HCV, several million infections. Why is this still happening? We have medicines, we have everything. That is because technology development and technology access, availability are two different things. This is the problem here. We still do not have broad screening campaigns. So we have the technology, but only less than 20% of the people are screened for these viruses today. That means a large majority of the people are going unscreened. And these treatments are good, but many people cannot afford them because they are expensive. Unfortunately, there is still no vaccine because of this high genetic variation. And that brings me to my last section, which is just 4 or 5 slides. I'm almost done. Where are we heading with this information? This is happening because of this important aspect. Biology research is very difficult. It's very simple to understand. Let's make a vaccine for HCV, coronavirus, but it's not that simple. That is because biology is quite complex. There are numerous parameters operating at one time, and net outcome is highly variable because of this region, have very little control. And plasticity, biology works. You do whatever. You give me coronavirus, I cut anything, still I can make that virus work. That creates problem for experimental design. But the advantage is biology requires, especially modern biology, multidisciplinary. There is no science that is irrelevant to biology. That I'm running out of time, otherwise I could have spent some time on this important point. Okay, so therefore, biology cannot be conquered by individuals. You need systems, you need teams, you need groups, you need countries to achieve this one. Therefore, biology becomes very charming at times. And the fight between immune system and microorganisms is eternal. Both are fighting each other for ages, each outwitting the other. There cannot be final winner unless we intervene with modern biology, with medical intervention, vaccines, medicines, and gene editing more importantly. So RNA viruses in this entire battle occupy a very unique position for multiple reasons. That is, RNA viruses come with many molecular strategies. All the RNA viruses use this very potent weapon, genetic diversity. They mutate very rapidly. Coronavirus is an exception to some extent. This is what happens. If you put one virus here, if there is no mutation, the dot remains a dot. But the virus mutates, therefore it spreads out into variant forms. This is HIV variation in one small African country, Congo, in a single year. Imagine what level of variation globally this virus can create. This is what all the viruses, RNA viruses do. HIV, HCV, HBV, influenza, coronavirus, all of them are doing this. This leads to the failure of vaccines, medicines, and immune system. The other interesting thing is here, every RNA is armed with own and unique molecular strategies. HIV uses this transcriptional silence a very powerful way to defeat the immune system. Influenza comes with genetic drift and genetic shifts because its genome is segmented. Coronavirus comes because it lacks this proofreading activity. It has largest genome and it uses host jumping as a very powerful strategy. I will not get into this because I don't have time. So therefore, RNA viruses are going to occupy an important part of medical research in the coming years. And for those people who are interested, there is so much of information available at these sites. And I'm done with my presentation. Thank you very much for your attention. And if you have any questions, I'll be happy to take them. Thank you.
Thank you, sir, for your excellent talk and sticking to the time limit. The floor is now open for questions.
I stop sharing this.
Yes, sir. Yes, Abhishek, you can ask.
Hello, sir. There was excellent talk. So I have a question regarding the last slide. So you told about the host jumping. Host jumping is very common in like influenza or HIV also even because like HIV transmits through the any body fluids and influenza also goes by like the aerosol way. So what is the special about this host jumping in the coronavirus?
Okay, yeah, that's a very good question. Actually, okay, what I mean by host jumping, I used a popular word. There is nothing like host jumping. Coronavirus is an infectious virus, respiratory virus, like influenza also. They are do the same thing. They are designed differently. They are acute viruses. They are designed to stay with the host not more than a few weeks, 1 or 2 weeks, not beyond. Unlike HIV and all other chronic viral infections, HIV is designed to stay with the host for a very, very long period, years and life, right? So therefore, the virus should be designed to outwit the immune system all its life. The virus comes with multiple immune evasion strategies, right? But whereas influenza and coronavirus don't have to worry about host immune system to that extent. By the virus is gone within 10 days or 14 days, not beyond. By that time, the host is not even making antibodies. So therefore, immune evasion is not such an important problem for coronavirus or influenza. Rather than that, what is important for these viruses is to move on, spread, expand from individual to individual. That's what they do the best. That's the reason they can establish very fastly expanding rapid epidemics in the globe. That's what I mean by host jumping. That's a very effective strategy. The immune system has no answer to this problem. That's what I mean by host jumping. Does it answer your question?
Yeah, Raju, you can ask.
Hi, so it's a nice talk. I just have a very simple and naive question, maybe. What I wanted to ask is that, as we know that the viruses are very much, mutating, and it is one of the causes is that because of their, fast replication and duplication strategy of viruses. What I wanted to know is that if it is so, then how does virus retain their virulence? Like
Are
if
you
there is more
talking about,
mutation.
sorry, are you talking about HCV?
I'm talking about viruses.
Oh, any virus, pathogenic viruses, you mean?
Yes, yes.
Yeah, see, mutation does, okay, let me put it this way. In a single individual, I will take an example of HIV. In every infected HIV infected person, at least there are 1 to 10 billion virus particles are made every single day. But most of these virus particles are defective because of mutations introduced all over. Let us say 99% viruses are dead viruses. That still leaves 10 million good viruses. See, this is the advantage the viruses have. The idea here is viruses want to make indiscriminately huge amount of genetic variation, even at the cost of some of these viruses not being good. Doesn't matter. Still, whatever that small tiny proportion good is sufficient enough for them. That is the trick, because they have no cost to pay for this variation. Gives a big advantage to them. So
Okay, I got it.
if the mutation is in a critical location, the virus is dead. Doesn't matter. There are many other good viruses.
Okay.
Any
is
other
this,
questions?
so I can ask actually a follow-up question. Is this like an example of evolution sort of happening in real time? It mutation is good for it because, even if some vaccine or something is able to control it, there are variants which can sort of spread. Is that
Yeah,
a way to think?
yes. Yeah, I mean, see, influenza virus and HIV are excellent examples for this. They are on the edge of collapse, genetic collapse. That's what they describe this as. They reach to the maximum, utmost maximum possible variation that is a biological system can create. So at one time, even scientists thought, can we push it a little more in the direction that the entire virus collapses? That concept looks attractive, but it did not work. So this is exactly the trick what HIV, influenza, and other viruses do. Create as much variation as possible, even to the extent that a large amount of virus is defective. Doesn't matter. Whatever still left is good enough. That is the trick. That is an evolutionary strategy, very excellent evolutionary strategy.
Hello. Yes, yeah. You can ask.
Hello. It's a
Yes.
very good talk. I also had a very nice question. So the like the infections, hepatitis infections, which are still undetected, because of which we have a lot of cases all over the world. Could it be possible because of some new like hepatitis virus version, which is also coming up, like something
Yeah.
else other than the ones that are
Yes, yes, I got
which
your
can be
question.
detected.
Yeah,
Yeah.
I got your question. I got your question.
Yeah.
See, right now, the most, you see, we are talking about 95% of infections. They are caused mostly by HBV and HCV. Now, are there new infections coming? That possibility always exists. You remember one of the slides I have shown you, there are 1.7 million viruses yet to be discovered. Even if one in 1,000 is really pathogenic, that is a huge number. So the possibility of a new virus coming can never be eliminated. That exists, but right now the problem here is we have the technology to detect both HBV and HCV, but we are not screening or we are not testing all the people. That is the problem one. Problem two is every test has its own detection limit. Don't forget that, right? So therefore, let us say influenza, you take coronavirus. And if this guy is infected yesterday, he doesn't have so much virus today. After 2 days, he will have. If I tested him one day after that, I probably will miss him because the test could not catch him because the virus is very small numbers in him. So every test also has a detection limit. So that also can limit screening capability. But right now the problem is less than 20% people are being screened. That is the biggest problem. So if we can screen all the people, then we can actually eliminate or make blood much more safe in the blood banks.
All
I
right.
have another question, if it is permitted, please.
Okay. Keep it short, Raju, please.
Yeah, sure. So it's a question of evolutionary point of evolutionary point of view. Uh, as you talked about coronavirus, and I just wanted to know is that because the infection to death ratio of coronavirus is very less. So
Sorry. Yeah, there is a
infection,
yes.
yeah. So infection to death ratio is very less for coronavirus or COVID virus. So what
Okay.
I wanted to know is that is it a strategy of viruses to balance their genetic load somehow?
I'm sorry, the question is not clear to me. You are saying that the infection rate is of coronavirus is small?
No, I'm saying infection to death ratio.
Oh, death ratio. See,
Yeah.
respiratory viruses are not killers. Don't forget that. I mean, influenza and all. See, the infection rate is so huge. The scale of infection is so huge. Therefore, it appears the death rate is small or big. But actually, these respiratory viruses are never infectious, are never killers. Their infection rate always is small. Especially this new coronavirus is not a big killer, unlike the previous coronaviruses. So every virus comes in a different shade and shape. They have certain things different. So this virus is not a big killer, but it infects very effectively because of its spike protein changes, mutations. I don't
Yeah.
know
So
whether.
that's what I my question is: are they changing? Just to exist in nature, like Do you think in future it will be like, it will not infectious, but they will still be like,
No. See,
coexisting?
there are some coronavirus that have become endemic, but I don't believe this coronavirus will become endemic, because it is, compared to those endemic coronavirus 4 viruses, this is very highly pathogenic relatively. A pathogenic virus cannot survive, especially a respiratory virus cannot survive in the population in an endemic form. It has to go. Whether it is going in 6 months or 12 months is the question here. Whether you have vaccines or no vaccines, this virus has to disappear. And in future, if at all there is a new coronavirus, that is not going to be this virus, it is going to be a new coronavirus. The reason is we have very huge, enormous amount of archived coronavirus library in the wild animals. So the chances of a new coronavirus coming is much, much higher than this coronavirus coming back. So this virus has to go, and it's never going to come back.
Thank you, sir.
Okay. Keeping time foundation and time, I request other people who have question to put your questions in the email ID that we are going to share with all of you in short time. And I'd like to thank Professor Ranga again and hand it over to Himara for the next session.
Thank you. My pleasure.
Thank you, Sheetal. I would like to introduce our next speaker, Honorary Professor D.N. Rao from Department of Biochemistry, Indian Institute of Science. He will be delivering talk in today's Nobel Lecture Series 2020 for the category of chemistry, which was jointly awarded to Emmanuel Charpentier and Jennifer A. Doudna for the development of a method for genome editing. Over to Professor D.N. Rao. Sir, please, you can go ahead.
I've just shared my screen. Can you see it?
No, sir.
No. You have to share again, Dian. Now?
Not yet.
Not yet, yeah.
Now.
Is it? It's there now. Yeah, you can go ahead.
Okay, all right.
Yes, sir.
Latest collaborations in biochemical history started in a cafe in Puerto Rico. It was in 2011, and Emanuela Charpentier and Jennifer Doudna were attending a microbiology conference that had a small session dedicated to a very obscure research field called the clustered regularly interspaced short palindromic repeats, or CRISPR for short. And from 2011, just 9 years later, the pair have shared the Nobel Prize in Chemistry. Although the two scientists did not know each other, they decided to explore the city together, that is in San Juan in Puerto Rico. And then they tried to figure out what were their interests in their respective areas. And they boiled down to the question was, what was this system doing in the bacteria? Now, before I actually explain the CRISPR-Cas system and what these two scientists did for the Nobel Prize, I just want to bring this point that in the past 100 odd years, 120 years, ever since the Nobel Prize was given in 1901, 110 prizes out of the 590 Nobel Prizes are in chemistry, and the most number of Nobel Prizes in chemistry has been awarded to scientists working in biochemistry, which are about 60 of them. These are the first, for a long time, when these four women, the Curies, Marie Curie, Irene Curie, and then Dorothy Hodgkin and Ada Yonath got the prize in chemistry. And 2 years back, Francis Arnold at Caltech got the prize for directed evolution. And now this year, we have Emanuela Charpentier and Jennifer Doudna, who developed a method for genome editing, but they actually repurposed an ancient bacterial immune system. And I thought I will mention about these bacterial immune systems. Earlier on, 60 years ago, another set of 3 scientists got the Nobel Prize for an immune system that they discovered in bacteria. Now, generally, as Uday Kumar Ranga also mentioned about the bacterial world and the fight between the viruses or the phages and the bacteria, the arms race as it's normally referred to. Phages are very small and they have to survive within the cells. They live, they multiply, they replicate and divide. So they have to absorb. The DNA is injected. Number 2 step, DNA is replicated, then transcribed and translated. Then individual phages are assembled. They kill the cell and the host cell lyses, and then the phages come out. So there are very many ways where you can stop the phage at various stages. You can prevent the phage absorption. You can block the phage receptor. You can the production of extracellular matrix proteins, production of competitive inhibitors, an abortive infection system, and finally preventing phage DNA entry. And that is where the 2 systems that I want to talk to you about, the restriction modification system and the CRISPR-Cas system. Few slides about the RM system. This was pioneering work of 2 people, Werner Arber and his scientific assistant Daisy Duswa. In 1962, a Swiss biochemist, Werner Arber, provided the first evidence for the existence of molecular scissors that could cut DNA. He showed that in a simple organism like E. coli, bacteria have an enzymatic immune system that recognizes and destroys foreign DNA, that is the bacterial virus or the bacteriophage DNA, and modifies its own DNA to prevent self-destruction, and which we call as the RM phenomena. And the basic role of the RM systems in bacteria is actually in evolution rather than in vital cellular processes. So it's a survival defense mechanism. Just like the eukaryotes or us have this immune system, this is a primitive immune system. So the host DNA is modified by methylation, which is now not cleavable by the host restriction enzyme, whereas the foreign DNA that comes in the form of the phage DNA is actually chopped off into pieces so that the virus doesn't multiply or the bacteriophage doesn't multiply. The discovery of Werner Arber, Daisy Duswa, and the published papers in 1960-61 led later on to the discovery of these systems in a number of bacteria, and 2 scientists from the Johns Hopkins University, Dan Nathans and Hamilton Smith, actually did a lot of biochemistry of these enzymes. And in 1978, the Nobel Prize in Physiology of Medicine was awarded jointly to Werner Arber, Daniel Nathans, and Hamilton Smith for the discovery of restriction enzymes, and we now know the application to problems of molecular genetics. The discovery of the restriction enzymes is the basis for all the genetic engineering, the recombinant DNA technology that we are all familiar, and therefore the potential of this discovery was recognized way back in 1978, and the Nobel Prize was given. There are lots of restriction enzymes, different kinds of restriction enzymes. The restriction enzyme generally recognize a specific sequence on DNA and cut within the sequence, as is shown here. But there are certain restriction enzymes which recognize the sequence, something like this, and cut away from the sequence, not from the recognition sequence, but not within the recognition sequence, but about 9 to 14 base away from the recognition sequence. A good example is an enzyme called FOC1, which actually recognizes very specific sequence, double-stranded DNA sequence, and cuts away from the sequence about 9 to 14 nucleotides away. So these are called the shifted cleavage because the cleavage is not at the site, but the cleavage is away from the site of recognitions. So Chandrasekharan Srinivasan, again from Johns Hopkins University, who was a postdoc in Hamilton Smith's lab, was working on the FOC1 and the system, and actually delineated the 2 domains in the protein called the DNA binding domain and the cleavage domain. And by studying the FOC1 restriction system, he actually developed some very, very nice editing, the first set of gene editing nucleases, where you fuse the cleavage domain or the cutting domain of the FOC1 to DNA binding domains of various proteins. This is a zinc finger protein, which is a DNA binding protein. And the DNA binding protein was fused with the cleavage domain of FOC1. And lo and behold, you have a new nucleus, which was recognized by the DNA binding portion of the protein a sequence, something different from what the fork-1 actually recognizes. So in other words, you could design restriction enzymes. And that is the first set of editing nucleases that the world had at its hand. And I think, I bring this point because the Nobel Prize this year is given to a method of genome editing. But the zinc finger nucleases were the first method. Of course, I know there are disadvantages of the zinc finger nucleases, but the whole concept was actually first described by Srinivasan Chandrasekharan, and later on used these by Dada Carroll at the University of Utah in Salt Lake to actually apply and do some very nice genome editing work. Following the zinc finger, this zinc finger nucleases actually came up in 1993-94. Following this, you had another kind of genome editing nucleases called the transcriptional activator-like effector called the tail proteins or the tail nucleases. And again, these are proteins that are actually found in a bacteria called Xanthomonas. And these DNA binding proteins were actually now again fused with the nucleus domain of the fork-1. And then you had the tail nucleases. This was much better than zinc finger because there was more specificity involved. And each of these residues here, which you've shown here, actually interact with these bases, the 4 bases, the A, C, G, T. So the zinc finger nucleases and the talons are the precursors of what we now have called these CRISPRs. And genome editing, as a brief definition for the first time, people who are listening to this area of research, is actually the objective of the experiment is targeted replacement of a mutant gene, which is shown here in red, with a normal gene shown in green. Okay, in a human cell, for example. In a converse experiment, one could replace a normal gene with a mutant allele or mutant gene, essentially knocking out that gene in the cells, and then you can find out what's the function of the gene. There are several genes which we don't know the function, and therefore this is one way of doing things. So knocking out the genes by using this is a very nice method. Oh, sorry, what did happen? All right. Genome editing is a process by introducing specific modifications in the endogenous genomic DNA, and by using homology-directed repair or non-homology end-joining repair, it can also be used for genetic correlation. So you have a protein-guided nucleases called the zinc finger nucleases and the talons. And now the CRISPR-Cas, which is the subject of the Nobel Prize this year, is an RNA-guided system. And I'll explain to you what this whole system is. But I must again come back to a little bit of history and what is there in the literature. In 1993, Francisco Mojica at the University of Alicante in Spain, as a graduate student, was looking at a lot of sequences. And he was looking particularly at the sequence of a genome of a salt-loving microorganism. And he found there were repeating palindromic sections of DNA separated by short spacer sequences. And he went back into literature and found, actually even before him in 1987, Ishido described a similar repeating DNA patterns in E. coli. But only when he became a faculty in the University of Alicante in Spain, that Francisco, and by just virtual correspondence with a good biochemist, Rud Hansen, coined the term CRISPR, which is the clustered, regularly interspaced short palindromic repeats. Even at that time, they didn't know what these sequences were doing. And only later on, they found out these were all part of a bacterial immune system. While the RM system is a good example of adaptive immune system, that the passive immune system, which we all know in eukaryote, is the new one that these people discovered. So CRISPR is actually, as it's written here, clustered, regularly interspaced, short palindromic repeats. And you have proteins associated with CRISPR called the Cas proteins. The CRISPR associated proteins called the Cas proteins, and they play in adaptive immunity systems. The CRISPR-Cas is found in most bacteria, 48% of eubacteria, and almost 95% of the archaea have this system. And this is how, what is the CRISPR locus, or the locus in chromosomes in bacteria. In some region, they have these kind of arrangements. And this is what I was talking about. You have repeat sequences alternating the spacer, which is written as 1, 2, 3, 4. And the repeat length is usually 21 to 48 base pairs, and the spacer length is about 26 to 72 base pairs. And the number of spacers vary from 1 to several hundred. And this is what Francisco Mojica actually found when analyzing the genomes of several bacterial genomes. And he didn't know what this were. It later on became very clear to him when he became an assistant professor that some of these sequences, that is the sequences in the spacer, actually coincided with a lot of sequences of phage genomes. So this was the first hint that these sequences in the spacers were actually memory molecules for the bacteria. These molecules actually came from the virus when it first attacked this particular bacteria. Now, I want you to be watchful when I'm just going through, because this is the heart of the whole work. First, I'm going to describe the whole system as a bacterial defense system, because this is what was exploited by Emmanuel Charpentier and Jennifer Doudna for the gene editing process. So when the phage first infects, for example, a bacterial cell, it injects its DNA. It injects this DNA codes for some proteins, Cas2 or the Cas proteins. And this is the fragment, the foreign fragment, which is acquired to the CRISPR locus. If the bacterial cell survives, then these small things here, the red ones, are actually incorporated. The foreign DNA fragments are incorporated in the CRISPR locus in the region of the E. coli genome, for example, with these repeat sequences. Between the two repeat sequences, the foreign DNA, the viral DNA, is actually encoded here. It encodes also the Cas genes, the Cas proteins, and it also has another very interesting molecule called the tracer RNA, which is actually discovered by Emmanuel Charpentier. And this is a contribution that earned her the Nobel Prize, because it was a very important contribution in understanding how this whole system works. So when you have the second infection, the second time the virus infects, you already have the viral portions of the viral genome actually incorporated in these spacers. So when you have a transcription, this long DNA molecule is now made into a long RNA molecule called the cRNA, the CRISPR RNA molecule, which is actually chopped off into pieces like this. And then the protein called the Cas9 and the tracer RNA molecule, which I told you that Emmanuel Charpentier discovered, all combine to form an RNA Cas9 complex formation. And this is what guides the Cas9 to the actual site where these sequences are present, to the sites on the viral genome. And the foreign DNA is recognized, and this whole triple-X structure, the tracer RNA, the CRISPR RNA, and the Cas9 are activated, and therefore the Cas9, which is a programmable, which is an RNA endonuclease, actually now makes the cut, and the foreign DNA is chopped off into a lot of pieces, and the bacteria become resistant to the virus. So this is the immune system. This is the adaptive immune system that I talked about. The RM system that Werner Arbut discovered is the passive immune system that was first recognized 60 years ago, and years later got the Nobel Prize. So this is the heart of the work. The repurposing of this whole system, of the bacterial defense system, is the basis of this Nobel Prize of Chemistry this year. One more slide, just to repeat that this work was done actually in Streptococcus species, which is a natural immune system. You have the viral DNA, which is shown here, the repeat sequences, the viral DNA, and then the phage infects, this DNA is injected here. The bacterium now inserts a piece of the virus DNA in the CRISPR section of the genome, and between each viral DNA is repeated sequence. The CRISPR DNA, this long one, is now transcribed to form you the RNA here, which is called the CRISPR RNA. And it is chopped off into pieces by the action of this RNase, the Cas9 and the CRISPR tracer RNA, which I mentioned that Emmanuel Charpentier actually, all these combined together, and in the presence of RNase 3, this is all chopped off into pieces, and then you have one whole unit called the genetic scissors, which has the Cas9, the tracer RNA, and the CRISPR RNA. And it is this that comes now and recognizes the sequence of the virus, because you already have a sequence, and therefore there's a homology search there. And then once that is found, once this, the base pairing occurs here, the scissors would activate and then cut the DNA. Simple as that. But it was not known for such a long time that another system, another immune system actually existed here. So the Cas9 protein is is a big protein, and now you have versions of the Cas9. You have smaller versions of the Cas9 protein, and a lot of work going on the structure of these proteins. And Jennifer Doudna's lab itself has done a lot of work on it. She comes from actually from an RNA background lab. She worked with Tom Cech and Jack Szostak, who got the Nobel Prize earlier for their work on RNA catalysis and things like that. So Jennifer Doudna is an RNA biochemist, RNA chemist, whereas Emmanuel Charpentier is actually a microbiologist, was interested in studying antibiotic resistance in microorganisms, and yet she was looking at all the small RNA molecules, the microRNAs, and the role of these microRNAs in this process of antibiotic resistance. But a combination of these two actually led to this whole discovery of genome editing, and they figured out that the bacterial immune system was a good system that could be exploited. So this is what it is. I just brought this particular thing, slide, just to show you that on the left-hand side, you have the bacterial system here, and now you have the CRISPR-Cas9 genetic scissors. While in the bacterial system, the tracer RNA and the CRISPR RNA are two different molecules. It is Jennifer Doudna and Emmanuel Charpentier who thought that if they combined these two molecules and made it as a single guided RNA molecule, as it's shown here, and you take Cas9, this two component system is enough to do cutting wherever you want. So you design that RNA molecule to a site where you want any kind of alteration, fix it with the tRNA, the tracer RNA molecule, and you have Cas9. These are all now available, the Cas9 is available commercially. The RNA molecules are much easier to synthesize. You can do it by, order these RNA molecules, and you can design a CRISPR-Cas9 genetic scissor the way you want it, and use it to wherever you want the alteration to cut. Once a double strand break occurs, then you have either the non-homologous end joining repair system or even the homology-directed repair system, which will actually correct, and then you have a new molecule that is formed. And this is what is the genome editing tool that people have talked about. This is, again, another slide to just show you what happens in the bacterial system on the left-hand side. And this is the in vitro artificial system that is now available here. So if you compare these three genome nucleases, editing nucleases, the zinc fingers, the talons, or the CRISPR, this is the best part of it. The treating efficiency, the targeting efficiency is very high in the CRISPR-Cas9, and therefore it became very popular. It became so popular that everybody now just completely forgotten about the zinc fingers and the talons. But there are some success stories with the zinc finger nucleases and things like that. So it's much easier. It is much easier, much cheaper to get a CRISPR-Cas9 system as compared to a zinc finger nucleus or the 10. These are all protein-based, and therefore any engineering we'll have to do in the proteins, whereas engineering with RNA molecules is the Cas9 is one protein. It is RNA guided endonuclease. And therefore, the issue of the other thing is that you could have, other than the target, there are cases of off-target cleavage, and this one has to be very careful, especially when you apply it to human therapy or food sources. And I think now various versions of Cas9 and various systems have come out with much and much less off-target frequencies, especially for the CRISPR-Cas9 system. So if you go back to this 20-year story of CRISPR, which started actually in 2000, actually in 1987, the Japanese scientists actually reported, but 2000 is when Francisco Mojica showed these sequences. And if we go now to 2020 is when the Nobel Prize has been given, it's been relatively short. Although people were studying for a long time, even within this 20 years on the bacterial adaptive system, it was only in 2012 that the paper that Jennifer Doudna and Emmanuel Charpentier published in Science to show that this works and that can be expressed and you can do gene editing, especially in prokaryotic systems. Just about that time, 4, 5, 6 months before, there's a scientist in Lithuania in Vilnius, Virgis Siksenis, a good friend of mine and Nagaraj also, working on restriction enzymes, was actually the first person the credit should be given for the whole mechanism of this Cas9, the CRISPR-Cas9 system that Emmanuel Charpentier and Jennifer Doudna. But his paper was rejected by Cell. It was not even reviewed. And then it took 4, 5 months, 3 journals, and finally published 5 or 6 months later in the PNA's proceedings paper. But by then, Jennifer Doudna and Emmanuel Charpentier had published it in Science in 2012. Just after this 2012 paper, I think at the same time on the East Coast, Feng Zhang and George Church had actually shown or demonstrated that Cas9 genome engineering is possible in eukaryotic cells. Whereas the Jennifer Doudna and the Emmanuel Charpentier papers talked only about prokaryotes and generally left it as other organisms. And this is the basis of the whole bitter patent war between the Berkeley group and the Broad Institute, the MIT Harvard group. And I'll talk to you about that later on. Whatever be the case, this is the Nobel Prize that was given for work at a very short span. Very quickly this was done. And why is there a case about the CRISPR system? Because it can be programmed to perform gene editing in mammalian cells. It's easier, it's simpler, and everything is and efficiency and stunning ease of use. In one sense, gene therapy is actually back here. I'll skip some of these slides just to show you lots of companies have come up. Both Emmanuel Charpentier and Jennifer Doudna and Feng Zhang, all these people working have their own genome editing companies, and you see the global markets for CRISPR-Cas9 have really gone up over the last, when it actually started in 2007 or 8, you see it's completely. The number of publications has shot up so much that it is shown in the PubMed. This is the number of publications, and patents have gone up, and there's a very bitter patent war between Feng Zhang of the Broad Institute and Jennifer Doudna at Berkeley. But I think now the court has clearly established and given the favor to the Broad Institute. In 2018, the patent was given to the MIT Harvard group, the Broad Institute group. So whether you do gene disruption by any of these methods, either by zinc finger nucleases, the TALENs, or the CRISPR-Cas9, the targeted double-strand breakers are finally repaired by all these systems here, which I talked about. And just to show you, there are lots of examples. You have white mushrooms that they will never oxidize at all. They resist browning. You have fantastic petunia flowers with beautiful colors. You have cows without the horns. That is possible because in many places this is not a favorable trait to have horns. And you have, for example, scientists have edited the wild-type dextral snail into a snail showing sinistral coiling here. development. There are lots of nice examples appear where simple genome editing by the CRISPR-Cas9 or even in some cases zinc finger nucleases have resulted in this. So whatever you want to do is possible. In ex vivo experiments where you take a patient with a disorder, for example, a blood disorder, a sickle cell or hemophilia, for example, sickle cell, I think that's most what they've done. You take the patient with blood disorder, isolate the CD43 and the other corresponding cells here, do the genome editing using the gene editing nucleases. Then you have the edited CD34 plus the HSPCs, and put them back, and you find in functional analysis, and you reinfuse into the human being. And a lot of some of these clinical applications have now come up in a big way. In the dairy industry, CRISPR immune system to protect important bacteria like Lactobacillus acidophilus, which is widely used in yogurts and dietary supplements, is heavily employed because of the phage infections that the dairy industry faces a lot. More recently, the CRISPR-Cas system, the CRISPR nucleases, both Jennifer Doudna's group in Berkeley and the Feng Zhang's group in MIT Broad have actually established a CRISPR-based diagnostic platform that combines nucleic acid pre-amplification with CRISPR-Cas enzymology for specific recognition of a DNA RNA sequences. They call it as Sherlock, and this is now heavily used in the COVID-19 detection in samples and things like that. And in India, we're not too far away, the IGIB in Delhi, with the help of the Tata Group, have actually developed another detective system they call as Feluda, but they've used another Cas9 from an organism called Francisella novicida, a very efficient Cas9, and they're actually using this system, Feluda, for detection of COVID-19. I think this is in the market now, and a lot of applications for this. So lots of applications, especially in the agriculture and plant-based research, the CRISPR-Cas system is being heavily used. But one has to always talk about this topic or the ethical debate around the CRISPR. When Jennifer Doudna published her papers, and years later on, in the morning, a Chinese biologist called her up, Jiang Qi, who said he had modified twin embryos using IVF, resulting in the birth of two girls allegedly containing alleles that would confer protection from infection by HIV. There was an uproar in the scientific community because human, germline manipulation was still not allowed. And Jennifer Doudna actually led the whole protest and said that genome editing of embryos should be stopped at, should be encouraged only where a clear unmet medical need exists. And actually, this Chinese scientist now is currently in the jail because of this work. So one has to worry about these ethical issues, especially when you're manipulating the germline, things like that. Two other things I want to mention here, especially in the CRISPR-Cas Nobel Prize of this year. While both Jennifer Doudna and Emmanuelle Charpentier did absolutely fantastic work, they deserve the prize. I think there are others who could have been included, but they who could have been included. Verghese Sixness, I told you about it 4 months before this paper came out of Science in 12. He had the whole mechanism that was published, but Cell refused it, and much later it was published in PNAS. George Church and Feng Zhang at MIT actually also was doing a lot of work on this CRISPR-Cas, and the patent, as I said, was actually given to the MIT group. What is more interesting, the three of them, Emmanuelle Charpentier, Verghese Sixness, and Jennifer Doudna, actually shared the Kavli Prize, one of the most prestigious prizes, $1 million prize. Then, George Church, Feng Zhang, Jennifer Doudna, and Charpentier shared another prize together. And the Nobel Prize, however, was given to these young, very bright scientists, one from Europe and one from the US. And I think especially in this year's prize, there's been a general talk about that the soaring popularity of the editing has made celebrities of principal investigators who pioneered the field, but the graduate students and postdocs are often overlooked. And George Church at MIT has taken a big view on this, and he's actually called them the unsung heroes of CRISPR-Cas. That one has to take the contribution of the first author on these manuscripts and give them some credit. This is Jennifer Doudna's lab, and she swears by this lab manager, whom she hired, Kai Hong Zhou, 20 years ago, and says the whole lab runs because of her and things like that. This is a book that Jennifer Doudna has written in 2014 or 15. I have this book with me. It's a nice book, A Crack in Creation, worth reading it. And I will end my talk by saying that at first glance, it may seem that the chemistry Nobel has been biologized again. It's sometimes hard to see how an enzyme or a phage are chemistry, but they are. It is chemistry that governs the shape of these molecules, which in turn affects their functions. And I think, the most beautiful thing that I've read about this prize, that both Doudna and Charpentier said that CRISPR is a perfect example of how solutions come from unexpected places. It's not about doing science for the sake of publishing and accumulating impact factors. It is about believing in a project that may not be as exciting or fashionable at first. The 2020 Chemistry Prize, one of the greatest collaborations, started in a cafeteria in Puerto Rico. It's a beautiful story that was extremely unexpected and came right out of the blue. And there's no other tool, as I know of, that has been so revolutionary in the hands of scientists and in the lives of others so quickly, 7-8 years after the whole system was actually described. So thank you very much.
Thank you, sir, for the enlightening talk. The floor is now open for questions. People feel free to ask questions. Shobik.
Hello. Very nice talk, sir. So I have one question. So you first told in the bacterial system the bacteria needs to cut the segment of the phage when it's first getting infected. So my question here is, what sets up that equilibrium, that if virus will win or the phage will win?
Yes.
Because it needs the time.
Right. The CRISPR locus has the provision to incorporate the viral genomes if the bacteria has survived the viral infection. If the viral infection takes an upper hand, there's no question, the bacteria is dead. But in many cases, you have not all the time the virus doesn't succeed at all, and a whole new set of mechanisms for this. You have lots of reasons that the bacteria have. You have something called the anti-restriction systems. You have anti-CRISPR systems, which now regulate when a viral will supersede in this infection. It's only when the bacteria survives an infection that you have these sequences, and this is a memory sequence.
Is there anybody else?
Yeah, I have a question.
Please go ahead.
Sir, that was a very nice talk. I have a very basic question. Is the CRISPR locus constitutively expressed, or is it expressed only when the bacteria encounters the virus again?
I think it is expressed, but I'm not particularly sure under all conditions or something. Maybe Nagaraj would know if he's there. I think
There is a low-level expression of. There is a low-level expression is reported, but I think actually most of the time this is a silent locus.
Right.
But when coming DNA, I think this creates a stress, and actually not much work has gone on to how these
is regulated.
CRISPRs are turned on, but there's an endogenous low-level expression of some of these Cas.
Okay, thank you, sirs.
Professor Munyappa, sir, one is raising hand.
You described type 2 Cas9 and Cas9 mediated engineering. What about the multi-domain or multi-subunit ones, like type 1, 3, and 4? Have
Yeah.
they any engineering kind of applications?
Very good question, because I don't know the answer. But there are for the multi-subunit proteins, the restriction enzymes. I've seen some reports about for the type 3 systems, but I don't know whether a lot of work has been done. I'm not particularly sure about it. The type 1s also have, yeah.
The type 3 CRISPR is equivalent to type 3 RF?
No, not at all. No, that classification is different. Type 1, the class 1 and class 2. It's a different kind.
But with all this, can you just highlight about the limitations of Cas9?
Yes, actually you have to put it into the cell now. How do you target the whole thing? Cas9 is a very big protein, and therefore, targeting is becoming a big problem. But now they've come up with a modified Cas9, some deletions and some mutations where they've created a smaller size. They found, therefore, there's a whole interest in searching for smaller Cas9 throughout the bacterial kingdom, and some work is going on. And I think they've been successful in getting smaller molecules of Cas9, which can get into the cell and be targeted. But there is a limitation, yes.
But what are these reports?
Too many questions you're asking.
No, I think you can't go away so easily. Reports saying that humans...
Give somebody else a chance.
I think they'll follow. This would be my final one.
Yes.
Because you've been talking about immunity. 50% of, in one kind of a study, where they found about 50% of people have innate antibodies for Cas9. Where did they get them from?
50% of?
In their survey, 50% of people were found to contain antibodies.
For Cas9?
Mm-hmm.
Interesting, yeah. I have no idea about that. It reminds me of this thing that I've read somewhere that, many of the people who require factor 8, for example, treatment in hemophilia, have antibodies against factor 8. So something like this. So it'll be interesting. Yeah, I have no idea about that.
Okay, so in the absence of any further questions, I would like to thank Professor D.N. Rao for this wonderful question-answer discussion session. Now, I would like to request Kshitija to end this wonderful session with concluding remarks. Over to Kshitija.
Am I audible?
Yes.
The remarkable discoveries in all the three disciplines are stories of ceaseless hard work, passion, and perseverance for all those who are and wish to pursue a career in science. What is absolutely amazing is that there is no stopping for the laureates. Professor Houghton and his team is working tirelessly on developing the vaccine and increasing its reach. Professor Andrea wants to make an even bigger telescope to observe the black hole, and the CRISPR team is working to use the technology for rewriting the genetic code for cells and organisms. With this, we thank our speakers, Professor Uday, Professor Rao, and Professor Prateek, for leaving us so much more informed and inspired. We thank the audience for joining this meeting and participating with such enthusiasm. You can drop your questions into our mailbox shown here. We will try to get answers for them from our speakers. Do follow our department's Twitter page and YouTube channel for updates on many more events and latest research. Until next time, stay safe and keep loving science.